Okay, so I welcome you again to another class. And in today's class, we have to be looking on toward another characteristic feature that we have for the eukaryotic cells. And they would be the cilia, and some of the very fewer cells, they also have the flagellum. Flagellum usually is a characteristic of the bacterial cells, but some of the cells of the human, they also have the flagellum in them. So what we know is that these cilias or the flagellums that we have, so they are the extensions of the eukaryotic cell, like they get out of the cell membrane. And overall, we say that they have a complicated structure. How their structure is complicated, we will see in a while. And you must know that these cilia and the flagellum, they are motile and they move in a rhythmic fashion. And when they would be moving in a rhythmic fashion, so this would be rendering some important role for the cells or for our human body as well. And if I were to tell you again, so as the centrioles were made up of the microtubules and the cytoskeleton was made up of the microtubules, so likewise the cilia and the flagellum that we have caught, so they are again made up of the microtubule. And we will see that how the microtubule they would align with an intention to be making the cilia and the flagellum. If I were to tell you about the composition that what makes the cilia and the flagellum, so they are made up of like some 600 polypeptide chains. We must clear our concept that what difference there lies between the cilia and the flagellum. If you go back to your previous classes, so we have got this male gamete, and this male gamete is the sperm cell. Okay, and the sperm cell would have a tail, and this tail was referred to as the flagellum. Okay, now if I take you to the respiratory system that you have learned in the previous classes, so you see that we have got these ciliated cells, and in these ciliated cells, we have got the cilia in them. So overall, if you just try to compare these two, flagellum and the cilia, so you see that the flagellum is fewer, like this is one per cell. And if I were to talk about the ciliated cells having cilia, so per cell, they are numerous in number. Their configuration overall is same for the cilia and the flagellum, but how frequent they are there for a particular cell, so that basically allows us to be calling them either the cilia or the flagellum. So the cilia, they would be numerous per cell, like we have the ciliated cells in the trachea and in the bronchus of our respiratory system. And if I were to talk about the flagellum, so they are again the extension of the eukaryotic cell, but they would be fewer, like there would be one or two extension per cell as we have got it for the sperm cell. But the important note is that the flagellum is commonly found within the bacteria, but in terms of the human beings, we see it in case of the sperm cell. Okay, now the function is that these cilias and the flagellum they would have a role in the movement or the locomotion. Like the flagellum overall is moving one cell in the female reproductive tract. And likewise, when I talk about the ciliated epithelium cells that we have in the lungs, and for the paramecium, which is again a kind of a eukaryotic microbe. So the ciliated cells, the cilia, they move, and that basically helps to move the mucus having the dust or the pathogens up the respiratory tract. I guess overall they have a role in terms of the movement and locomotion. So now I would like all of you to be very much clear about understanding that how come this cilia would be formed. Now, here I have made the diagram of the ciliated cells that you have got in the trachea and the bronchus of your airways or of your respiratory system. Now the situation is that here what I have with me, so this is the cell membrane, okay, and the cell membrane is having an extension out of the main body of the cell. And within this extension of the cell membrane or the cell that we have, so we would have these microtubules, and these microtubules they would be making the cilia. And at the bottom, these cilia they would connect themselves to the basal bodies, and you know that these basal bodies they are actually the centrioles with us. So the normal situation is that these centrioles that we are referring to them as the basal bodies at the basement of the cilia, so they would assemble the alpha tubulin and the beta tubulin, and they would make the protofilaments out of them. And those protofilaments they would move outside of these basal bodies, and as a result of that, we would make the cilia structure out of it. So we have to be very much clear that how come the microtubules that we have in these extension they would have a configuration, and how come the microtubules that we have or that are making the basal bodies they would have the configuration. So the basal bodies have the same structure as of centrioles. So if I were to tell you about the configuration or the structure of or the arrangement of the microtubule that we have in the basal body. So again, what I need to do is that I would take total of 27, you could say, microtubules. And these 27 microtubules that I have got with me, so I would convert them into total of 9 triplets of microtubule. So those 9 triplets of the microtubule, they would make one basal body. The other 9 triplet of the microtubule, they would make another basal body. Now, if I were to take you to be understanding that how does a microtubule, they are arranged within these cilia that appear as an extension and they get out of the basal bodies. So you see that here is an extended diagram of that. So the situation is that within these extensions, we would have total of surrounding 9 duplets, and at the center we would have 2 duplets. So these cilia, they have a configuration of the microtubule that is termed as 9 plus 2. 9 mean 9 surrounding duplets, and the 2 mean the central duplets, and they are enclosed by the cell membrane. If I were to count these microtubules for you. So here you see, if I zoom it more, so I have got here is a duplet, okay? So this is one duplet. And likewise, this is second, this is third, this is fourth, fifth, and this is sixth, seventh, eighth, and then ultimately we have got 9 duplets. And then within the center, we would have these two central duplets, central microtubules. Now, if I were to talk about that, how does the configuration of this duplet of the microtubule would look like? So this duplet further has got two microtubules. Like we would have the A microtubule, we will have the B microtubule. The B microtubule that we have over here, so we call this as an incomplete microtubule because in the B microtubule we don't have 13 protofilaments but rather than 10. Because what we know from before is that when we would have 13 protofilaments that make one complete microtubule. But since the B has got 10 protofilaments, so this would make an incomplete microtubule. For the A that we have, so it would have total of 13 protofilaments, and the 13 protofilaments, they make one complete A microtubule. The same microtubule further has got the side arms, and these side arms, they are made up of a protein, and the name of the protein is referred to as the Dynein protein. So if I were to summarize all of this for a while, so the basal bodies, they have got 9 triplets of microtubule. The cilia that we have, they have got two surrounding 9 duplets, two central microtubules. The 9 duplets that we have in them, again, we would have the A and the B microtubule. And among them, the A is a complete microtubule having 13 protofilaments. B is an incomplete microtubule having 10 protofilaments. And collectively, the basal bodies and these 9 plus 2 configuration of the microtubule along with the cell membrane. So this makes a complex, and the name of the complex is entitled as exoneme. Now we have to be clear that how come the micrograph of these cilia say would look like. So if you just focus here, so in the photomicrograph, what we see here, so this is the cell membrane. At the center, we have got two central microtubules. And here we have got here at this point, so they are the duplet of microtubules. Now among them, like if I were to zoom further, so you see for a moment that here we have got these side arms of the A microtubule, which is a part of a duplet. Okay, and then here we have got this A B microtubule attached to the A microtubule, and here you see at the center we have got two central microtubules as well. So I hope you would be clear with the structure of this cilia for now.